It's using the page down.
Yep, using those up and down arrows on the left side of the screen.
Oh, that's right. Okay, got it.
Yeah. Well, so before we get started today, and I guess we're a pretty small audience for now, I'd like to remind everyone that our next scheduled webinar was to be November 20th with presentations by 2 TCAP graduate students. We have not gotten any volunteers for that yet, so I guess stay tuned as to whether that's going to actually happen or not. Keep an eye on your inboxes. Of course, if you'd like to volunteer to present or volunteer somebody else to present, you can send me a message. But our webinar speaker today is Dr. Craig Morris. Dr. Morris has been the director of the USDA ARS Western Wheat Quality Lab in Pullman, Washington since 1989. He received his undergraduate degree with a double major in agronomy and seed science from Iowa State University in 1982 and went on to obtain a master's in crop physiology and a PhD in agronomy from Kansas State University. Dr. Morris is also an adjunct professor in the Washington State and University of Idaho School of Food Science and is currently on the board for the AACCI, which I guess is the international version of the American Association of Cereal Chemists. Is that correct?
Yeah, I'll mention the name change here in a second.
Okay. So the Western Wheat Quality Lab conducts cooperative investigations with plant breeders, geneticists, and pathologists in 7 western states, including Arizona, California, Idaho, Montana, Oregon, Utah, and Washington, to evaluate wheat breeding lines for milling and baking quality characteristics. It also conducts basic research into the biochemical and genetic basis of wheat quality and develops new methods to assess quality potential in wheat cultivars. So today Dr. Morris is going to present a lecture titled Wheat Grain Composition, what it is, how we measure, I guess, uh,
No problem.
well,
I
this
changed the title on you.
changed the title on me.
Sorry
Okay,
about that.
well, I guess I'll let you get started.
Okay, great. Thanks, Sarah. Yeah, I guess I had forgotten that I had actually sent you a title, but either way, it works out. So this is actually something I sort of ad-libbed from what the sort of the TCAP statement was. So I thought it would be more fun to integrate breeding and genetics into crop improvement and utilization. So anyway, the original title also is apropos. And just thanks, Sarah, for the nice introduction. So the thing about AACC International is since its inception, it was called the American Association of Cereal Chemists, which would be my primary professional affiliation. And then a few years ago, they've changed the name to AACC International. Since then, nobody really knows what they're talking about. But anyway, I've been quite involved in that organization and also a member of crop science for, I hate to admit it, 31 years. So, okay, so the Western Wheat Quality Lab is one of 4 regionally supported wheat quality labs run by the U.S. Department of Agriculture's Ag Research Service, and you can see where beautiful Pullman, Washington is located there on the map. The others are essentially geographically located based on wheat production. So we've got a lab up in Fargo, North Dakota that handles the hard red spring and durum wheat, one in Manhattan, Kansas that handles the southern plains, the hard winter wheats, and one back in Ohio that handles the eastern soft wheats. So this is my building. We're located on the Washington State University campus, which is really an excellent situation. And you'll find nationwide that many, if not most, ARS scientists are actually co-located at land-grant universities. So it's a good system. So getting into the talk, and this is tough for me because usually I like to have interaction with the audience and ask questions and get some feedback and stuff, but we'll have a go at it this way. So first, let's just sort of start exercising our minds. Why do we grow crops? and I came up with at least 5 of these. There are certainly more, but they didn't start with F, so I didn't list them. Food, feed, fiber, fuel, fertilizer. Some of those you may find a little bit unusual, but for example, obviously fiber, we're growing cotton and flax. Fuel, we can see that both for biodiesel as well as fuel ethanol, we're growing a lot of crops for fuel. And then fertilizer, you might not think about it, but certainly to some extent now and absolutely in our past, we grew a lot of leguminous plow-down crops specifically to as a source of fixed nitrogen for fertilizer. Much less so, I guess, now in these modern era. I would like to now focus us from crops down to grain crops. And my definition of grain crops is basically anything where we are producing and harvesting dry seeds. We can further subdivide that into certainly cereals that we would all recognize as grain crops, but also the pulses, oil seeds, and then a few crops that we refer to as pseudo-cereals. These would be things like quinoa, buckwheat, again, dry seeds, but don't really fall into any of the other 3 categories. So, breeders, I know this TCAP thing is primarily focused on breeders. So are breeders working for the farmer? Are they working for the consumer, or both? Well, we also need to think about, for example, the baker, the brewer, the miller. And I like to adopt the philosophy of we're working really from the farm to the fork. Well, everything that we're concerned with is in this little package, and whether it's a wheat kernel or a maize kernel or some other crop seed, it's basically all here. Absolutely, composition is very important, but we also need to consider some of the physical properties of the grain, and I will also suggest we need to consider a few physiological properties of the grain, and I'll elaborate on that here in a minute. Well, this is pretty standard. Everybody, can regurgitate this. Composition and physical attributes result from G, E, and a G by E interaction. But we also need to include and at least think about post-harvest processing. And in my mind, I lump everything that's not purely genetic under environment, and that can be climate management, soil, pathogens, et cetera, et cetera. So in terms of, I guess, our cereal grains, and in terms of crop quality, I've made sort of a general ranking here of what I consider to be more complex to less complex. And maybe it wouldn't surprise you that I've put wheat as, in my mind, the most complex in terms of end-use quality and utilization, followed probably fairly closely by malt barley and rice. Oats, certainly a lot simpler. And then when we get down to maize, feed barley, and grain sorghum, I'm sure there's some difference of an opinion, but really very little in terms of actual end-use quality. We're basically going for yield. We may consider something about protein content, but really there's not a whole lot to it. Well, U.S. statutory law recognizes 8 classes of wheat, and these being durum, hard red spring, hard red winter, soft red winter, hard white, soft white, and then a couple that you rarely hear about, one that's called unclassed and one that's called mixed. And if you want the exact definitions of those, you can look up through the Federal Grain Inspection Service and see what those are. In addition to these 8 classes, 3 of the classes actually have 3 subclasses, and those being durum, hard red spring, and soft white. So we've got quite a few types of wheat, and we need to think about why these are. Now, in addition to all these classes and subclasses, I would say nearly all wheat is also bought and sold on a minimum or a maximum protein specification. And like other cereals, we certainly have grades. For example, US number 2 hard red winter wheat. So again, why so many types of wheat? Well, the answer lies in that wheat is used in a nearly limitless variety of foods and is certainly the leading cereal in terms of human consumption. That's globally. So all of the sort of, pan breads, hearth breads that we would have some familiarity with. Also around the world, a lot of what we refer to as flatbreads, pita breads, pocket breads, naan, all this kind of stuff. Throughout the world, lots of different types of noodles. Now, we differentiate noodles from pasta, noodles being made primarily from hexaploid wheats, pasta being made from durum wheats. You know, we primarily think of these as being associated with the Orient, but certainly through Western culture, we have adopted and enjoy a lot of different types of noodles. Certainly soft wheats, all the cakes, cookies, pastries, this types of things. And as I mentioned, pasta originating primarily from durum. So the other thing we need to get our heads around is there's generally no absolute definition of quality, unlike yield or, say, disease resistance. Everyone can understand that given, say, two varieties, one yields more or one has a higher disease resistance. Well, in the case of quality, there's no sort of absolute definition. We would not think of, say, gluten strength as being an ultimate definition. We may want stronger gluten or we may want weaker gluten. And so really when we think about quality, we need to think of it as suitability, and the suitability is defined by a particular use, more times than not a particular food. The other thing is in terms of plant breeding, we really need to equate quality with the phenotype. So when we think about quality, whether it's a cookie quality or milling yield or what have you, that is basically the phenotype that we're working with in our cultivars and plant breeding populations. This is kind of how I imagine that the connections exist. It should start with the consumers, and that may be more philosophical than real, but certainly the type of foods that you and I choose to eat, the foods that we enjoy and will buy at any given value or any other preference, really drives back through food companies who are buying flour, so we have to turn wheat into flour. We need to include the grain trade because certainly in complex economic systems, the buyer and seller rarely are face-to-face. So the grain trade is important for the orderly marketing of wheat. And then the primary producer, which is farmers or farmer groups. Most in the United States, of course, most of the major wheat producing states have wheat commissions, have grower groups that represent farm interests. That should all funnel then into plant breeding and genetics, and whether you're a pathologist or a serial chemist, I consider those people to be essentially members of the same plant breeding genetics team. Regardless of whether you're actually the plant breeder or not, the team is working together to ultimately develop wheat varieties with improved traits. And of course, in my interest, it's end-use quality traits. Well, one thing we need to keep in mind is that plant breeding is a pretty tough job, and this gives you a general sense of the success rate of at least wheat breeding. If I look around at my friends that are wheat breeders and we get an estimate of the number of hedgerows that are being grown, a very, very tiny proportion of those end up being released as cultivars to farmers. Okay, let's get into some of the major end-use quality traits of wheat, and I'm going to go through these kind of in chunks. The first set here basically relate to the kernel, both on a, as you could think, an individual kernel basis as well as a mass of grain. So for example, and by far the most important in wheat is kernel texture, or we also call it kernel hardness. Since we out in the Pacific Northwest grow primarily soft wheat, we like to think of it as texture, not hardness, because obviously we could also have kernel softness. But kernel color is important. Red wheat, white wheat, kernel shape. Actually, we wouldn't normally think about shape, but shape is the primary morphological criterion to classify spring wheat from winter wheat. So it does have some importance. Kernel size, not probably super highly heritable, but certainly millers want larger kernel wheats. And as I mentioned, in mass, test weight or bulk density is an important trait. So as I go through these different sets of quality traits, I'm going to try to give you some illustration of what they are, why they're important, and to some extent, without going into any great depth, how we measure them. So the first of these, again, kernel texture, and we have two major classes throughout the U.S., hard wheat and soft wheat, and that is independent of whether those are spring wheats, winter wheats, or red wheats or white wheats. Kernel texture is certainly the single most important end-use quality trait in wheat, and you can see that it really affects everything from, in this case, cookie quality to milling and flour properties. At a very fundamental microscopic level, we know that hard wheat endosperm and soft wheat endosperm are very, very different. They behave quite differently and have quite markedly different material properties. In soft wheat, the starch granules, as you can see up there in the quadrant labeled B, the starch granules are very loosely attached to the protein matrix, and during milling, fractures occur typically around the starch granules, and a lot of free starch granules are released. In D, on the lower two panels, you can see a very hard vitreous endosperm. And in this case, the starch granules are embedded and cemented so tightly in the protein matrix that during milling, fractures often occur through starch granules. So it's a very different kind of process. In general, we would use this instrument. It's called the Perton 4100 Single Kernel Characterization System, or as we refer to it, the SKCS. This would be really, it's become sort of the industry standard for measuring kernel hardness. Down in the lower left, you can see a black wheel that turns, and just below it is a crescent with a progressively tighter gap. And the instrument drops individual kernels in that. As the black wheel rotates, it eventually crushes the kernel, and that crescent being attached to a load cell records and translates that crush profile into something that we recognize as kernel hardness. The nice thing about this instrument is it also measures each kernel's weight, the moisture content of each kernel, as well as an assessment of its diameter. And you can see up in the left-hand corner, it actually produces in real time a histogram of as it's crushing the kernels. And it also then, of course, outputs a summary of those data. At a genetic level, and this is really the genes that we are breeding for when we're selecting hard and soft wheats, the basis for kernel texture are the pyroinulins, pyroinulin A and pyroinulin B. In this case, I'm showing a cartoon of the primary amino acid structure of pyroinulin B. The boxes around the outside with arrows are actually indicating individual single nucleotide polymorphisms, each of which converts pyroinulin from a functional soft endosperm causing protein to a hard dysfunctional. So let me say that again. So basically, if you have functional pyroinulin A and pyroinulin B, you have a soft endosperm. If you alter either one of those genes through mutation, you end up with hard kernels. And this is just listing a few of the most of the hardness mutations that we've at least currently know about in hexaploid wheat. So the interesting thing about pyroinulins is that it provides a nice example of what I think of as a genetic cascade for quality. So we start out, of course, at the gene level. We have a purulin haplotype, which is either going to be hard or soft with a specific pin A and pin B gene sequence. Naturally, that leads to expression of the proteins, which in turn, although we don't know exactly how, conditions the kernel texture to be soft or hard. That in turn has a profound effect on milling performance and affecting things like flour starch damage and the granularity of the flours. That in turn has an effect on dough-water relations, how much water it takes to make an optimally formed dough, and then sequentially into processing and baking performance, and ultimately the end product quality. So it's a nice connection of where we actually have a stepwise connection, as it were, between the gene and ultimately end product quality for the consumer. So the next area of end use quality traits I kind of lumped together is basically milling performance. There's a lot of aspects about milling, and certainly we're not going to go into depth in them today. I will say, if you're not familiar with wheat, we mill wheat at a moisture content higher than is safe to store wheat. So normally whether it's corn or wheat or what have you, most cereal grains cannot be stored much above about 11 or 12% moisture at typical temperature ranges without running the risk of mold growth. However, we mill wheat at anywhere from 14 all the way up to sometimes 17 or 18% moisture content, depending on the hardness and the type of wheat. That water is actually added to the grain, typically anywhere from about 12 to 24 hours just prior to milling. The reason we add water, it's called tempering, is to toughen the bran, because we want the bran to hold together, and it also mellows out the endosperm, making it more friable and easier to grind. So the next aspect is what we refer to as break release, or sometimes break flour yield, and that really has to do with how the endosperm, as it were, comes apart when you apply pressure in the flour mill using steel rollers. Obviously, it's a grinding and shearing effect. We basically want to take the kernel, we want to separate the endosperm, we want to grind the endosperm down to a very small particle such that it can be construed as flour. Flour particle size is an important aspect, primarily controlled by kernel hardness, but obviously the way we mill wheat also has an effect. As I mentioned earlier, whenever we break a kernel or break a piece of endosperm, we may encounter some level of starch damage, and again related to hardness primarily. Just like the farmer wants high grain yield, a miller wants high flour yield. The more flour that they can produce per unit time and per unit wheat grain, the better. That's how they make their money. Bran cleanup refers to the phenomenon of where we actually want to separate the endosperm from the bran. And those are two separate botanical tissues, and the more cleanly that those two will separate from each other, the better, because mechanistically we would like to have all the bran in one place and all the endosperm in another place with a perfect separation. Ash content relates to the fact that most of the mineral content in a wheat kernel is in the bran. We don't really so much care about ash content per se, but it does serve as a proxy for bran contamination, or the amount of bran that has been ground up and has ended up in the flour. And then lastly, color. In the West, most people want white flour. They don't want gray or slightly dark flour, nor do they typically want yellow flour. If we look around the world, there's plenty of instances where consumers prefer a pale, creamy yellow color, and we all have experience with durum, where we have a very high carotenoid content, where the yellow color is actually one of the leading quality factors. So this slide is simply showing the 4 different milling systems in my lab, and they aren't unique to the Western Wheat Quality Lab. Starting up in the left, there's what we call a micro mill. This was designed at WSU, and this really was aimed at being able to mill a very small sample of wheat, anywhere from on the order of, say, 10 to 30 grams. Doesn't have a lot of milling action, but again, it's amenable to a very small sample. The next one over is a Buehler mill in the white in the middle. This would be typical of if you go to a commercial flour mill, this would be in their laboratory. This would be a small laboratory mill. For us, it's fairly large. It's really, in a sense, too big and too slow to run breeder samples. Typically, we'd run about 1 gallon of grain through that mill. The next one over is our pilot mill. This mill, as you can see sort of from left to right, there's little, looks like channels, and each one of those is indeed a set of steel rolls, each one effecting a different grinding operation. This mill has a very nice emulation of commercial milling, but it also needs about 100 pounds of grain to really get it fired up. So we would use this at the very last stages of cultivar development in our industry trials, where we actually have a very few number of the most elite breeding lines, and we're actually wanting to see how they perform in a commercial setting. On the bottom is the milling system that we would do nearly all of our breeding samples through. And in my lab, we would typically run a half kilo, 500 or 600 grams through this system. It uses 2 different grinding operations and 2 different sifting operations. So not only does it produce enough flour for all of the subsequent testing, but it also gives you a very good assessment of the milling performance of any given line, breeding line or cultivar. This instrument is automatically measures moisture and ash content of flours. Again, we have to know the moisture content so we can run our subsequent tests. Again, the ash content is a proxy for bran. And you can see over there on the lower left what typical wheat bran looks like coming out of a mill. If you look closely, you can kind of see that it's somewhat whiter, snowy white on one side compared to the other, and that's simply the last final vestiges of endosperm still stuck to the bran. And again, the miller would like to scrape away every last little drop of endosperm so that they can sell it for flour. But the competing force is that every time you grind it, you're going to fragment the bran, and some portion of that is going to be fine enough, small enough particle to end up in your flour, which generally is not desirable. Even though we like to eat a lot of whole wheat bread, there's still a lot of white flour that's produced. So the next topic is protein content as well as gluten strength and amount. Now these are intimately interrelated. Protein content, protein is fairly easy to measure, but really what we care about is not protein per se. What we really care about is gluten strength and the amount of gluten, because that's the horsepower that drives the car. And again, in some situations we want strong, high amount of gluten. In other situations we want a moderate amount, and then in still further situations a very low amount of very weak extensible gluten. And that all depends on the product we're trying to make out of it. So you may have heard the word Keldol nitrogen. Very few places actually run Keldol anymore. It's a nasty, nasty assay. It uses hot sulfuric acid to hydrolyze all of the proteins into elemental nitrogen. This instrument actually burns the sample. It's a combustion nitrogen analyzer, and it measures the amount of nitrogen in the sample, which we then convert using a conversion factor of 5.7 to protein. And again, this would be the international standard for measuring protein content of wheat. We also use quite a bit of near-infrared reflectance spectroscopy. This is an NIR protein instrument. There's a wide variety of types, sizes. Some use whole grain, some use flour, some use ground grain. Good instruments, not real complicated, but they do not record or measure anything fundamentally about the nitrogen or the protein. So every NIR instrument has to be calibrated against elemental nitrogen using the, in our case, the combustion instrument that you saw just previously. So what's the big deal about gluten? Well, since most uses of wheat involve bread or bread-like products, the driver behind that is gluten strength. And gluten forms elastic networks, which you'll see here in a second. Two aspects of gluten and protein. One, that certainly on the top we see that generally speaking, as protein content goes up, bread quality goes up. And that's because we're adding more gluten as we increase protein content. And that is why also there is almost always a significant price differential among our different market classes of wheat. Hard red spring, high protein, almost always being considerably more expensive than, say, hard red winter, and more expensive than soft white. Now down the lower left-hand corner, you can see what we conceptually think of as gluten quality. So both of these flours are at 12% protein, but they make dramatically different bread. So that gets back to this whole aspect of genetically, the composition of the gluten is what really drives product quality. On the lower right, you can see that as cereal chemists, we can separate the storage proteins of the wheat kernel into two general classes, glutenin and gliadin, and you can see that they have different properties in terms of their viscoelasticity. When mixed together, they give a balanced product, which we recognize in term gluten. And again, gluten proteins have this unique ability to form elastic networks. If corn could do this, and under certain conditions it can, we probably would be making pan bread out of corn, but we don't. We make it out of wheat. And it's not because we designed wheat this way. It's because wheat has this inherent ability that it's the storage proteins in the kernel can form these elastic networks. And obviously, humankind has taken advantage of this property and used it as one of our leading food sources. As I mentioned, for bread, gluten must form viscoelastic doughs because really it's that viscoelastic dough, or we can almost think of it as a sponge, to hold the gases that are produced during yeast fermentation. So we have primarily carbon dioxide. Of course, ethanol is produced during yeast fermentation, same as in beer. And the properties of the gluten, the properties of the dough, have to be such that they can trap that gas and expand without the bubbles, the gas migrating to the surface of the bread or whatever the product is, the gas escaping and the product collapsing. Now for soft wheat, usually we want gluten to not contribute to poor texture. So we don't want a tough, chewy, or hard textured cookie or soft wheat product. So that's an example of where we don't want gluten to interfere with it. This is very sort of conceptual, but it gives you some sense of the range of gluten strength for the different market classes. Now I haven't mentioned club wheat, but club wheat is a type of wheat grown exclusively in the Pacific Northwest, at least in the United States, a little bit grown in Australia and other places around the world. It is by design the weakest gluten strength wheat produced in the United States. So if you're looking to make some very light confectionery product where you absolutely don't want any gluten development, you don't want any gluten to interfere with product texture, club would be the thing to go for. Soft white typically has quite a wide range in strength, from very weak to getting considerably strong. And I think probably hard red winter, hard red spring, as well as hard whites are illustrated there. Again, hard spring typically having the highest gluten strength. Well, how can we measure gluten strength? Well, one of the easiest is something called the SDS sedimentation test. SDS, as most of you know, is sodium dodecyl sulfate. It's basically a detergent. It has the ability to swell gluten proteins and form almost sort of gels in some sense. And in the presence of SDS and aqueous solution, the different glutens swell base, and it correlates with essentially their baking performance. So the gluten proteins that tend to swell more are also the ones that tend to bake better bread, and conversely the ones that are weaker tend to have lower SDS sedimentation volume. So it's a quick, easy test for initial screening of gluten strength in wheat. So one instrument that we developed in my lab is a laser-based instrument to try to measure in real time the sedimentation of wheat grains, whole grain meals. Basically, instead of turning it into flour, we would actually take a sample of wheat and just grind it into a meal and put it in these tubes. And as the material settles in the SDS solution, the lasers with diodes on the other opposing side of the tube measure the sedimentation phenomena in real time. So the next area is starch, and of course the leading component of a wheat kernel and the endosperm is starch. Anywhere from, say, 65 to 75% or more of the wheat kernel is starch. There's several things that are important to us in terms of cereal chemistry and product quality related to starch. Those being what we would refer to as pasting and also gelatinization. Both of these phenomena require water, but I would be hard-pressed to think of any food that we make out of wheat where we don't have some significant level of water. Both are thermodynamics phenomena. So in the presence of water, when we heat flour, the starch granules gelatinize, and also a characteristic that we refer to as pasting. Differences in pasting and gelatinization are primarily affected by the amylose-amylopectin content of the starch. These two molecules are under different biosynthetic control. They're in different biosynthetic pathways. They have different enzymes that create them, and can be independently genetically manipulated. And then in terms of a test, we have something called flour swelling volume, which captures another attribute of starch, and that is the extent to which the granule will swell when heated in the presence of water. And different starches, based on their amylose-amylopectin content, swell to different degrees, and I'll show you a picture of that here in a minute. So wheat starch granules come in two general sizes, referred to as A granules and B granules. The A granules are much larger, lenticular in shape. Almost every cereal, whether it's rice or maize or wheat, has fairly unique characteristics about its starch granule morphology and composition. The enzyme and gene that we would be most interested in terms of starch composition would be granule-bound starch synthase. Granule-bound starch synthase, or GBSS, which is a lot easier to say, is responsible for synthesizing amylose. And as we routinely encounter in hexaploid wheat, we have a homologous series of 3 genes located on chromosome 7A short arm, 7D short arm, and then on the translocation for chromosome 4A long arm. And in the lower portion of the picture is a photograph of an actual stained polyacrylamide gel electrophoresis, showing the 3 proteins on the far right that you can just barely separate, and they're all approximately 60-some kilodaltons in size. So we can look at the presence or absence of the proteins in gel electrophoresis. That's how we did it for many, many years. Of course, now in the era of PCR, there's a lot easier ways to genotype breeding lines. In some sense, the phenotype is actually so dramatically different based on GBSS haplotype that we don't even really bother about doing any genetic work. We simply run the samples through, in this case, FSV, which is this flower swelling volume. And if you look at the test tubes, those are labeled according to how many waxy mutant alleles they have. So 0 means that you actually have 3 functional copies of GBSS. I'm sorry, that's just opposite. You have 0 functional copies of GBSS, therefore you have no amylose, and you have the greatest amount of swelling. So the starch actually swells in hot water and will completely fill the entire volume of the tube. On the far right, we have 3 functional copies of GBSS, so we have the maximum amount of amylose that would be typically produced. That starch has the lowest amount of swelling. On the bottom, we see an instrument called the RVA, and you can see there sitting on top of the RVA is an aluminum can with a little paint stirrer that's actually upside down. We would put some flour and water in that. It goes into the machine, it starts to stir it and heat it at the same time, and it continuously records the viscosity of that flour water mixture as it gelatinizes and pastes. Just to the left of the RVA, then we actually see the viscosity of those gels or those pastes. And as related to the swelling, as we reduce amylose, we increase what we call paste viscosity. If we knock out all 3 GBSS genes, we end up, as I mentioned, with no amylose. It's what we call waxy wheat. And at this point, we have flours and starches that behave very, very differently from normal wheat. And in the upper right-hand corner, the white opaque, almost looks like a scallop or something. That's normal wheat starch, and it forms a fairly rigid gel when pasted and then cooled. Just to the left is what waxy starch looks like, and without the amylose, the gel actually has very little structural integrity. So the next area of end-use quality of traits in wheat has to do with the non-starch carbohydrates. And in wheat, we're primarily focused on arabinose islands, or what we used to call pentazans. And these are very analogous to what we might have heard, say, in barley and oats, which is beta-glucans. Wheat has very little beta-glucan. Instead, it has arabinose island, but they're very similar types of non-starch carbohydrates. Another area which I can't say I personally know much about is fructans. So in terms of fiber in wheat, we're really focused on arabinose islands. Now, in addition, I'll just interject that we're certainly interested in pigments in wheat, and especially the carotenoids, primarily related to durum, wheat, but I would remind us that, you know, we have red wheat, red bran wheat, we have purple wheat, blue wheat. So a lot of these pigment systems that we would not normally encounter are pervasive through the plant kingdom, including wheat. Here's a illustration of the arabinose primary structure, comprised of arabinose and xylan moieties in a large polymeric structure. They have the unique ability to form cross-links, and on the upper panel we can see that the ferulic acid that's covalently linked to arabinoxylan can form cross-links, and as well the arabinose through ferulic acid can form cross-links with tyrosine moieties on proteins. And of course, as soon as you form even just one cross-link, you've effectively doubled the size of the polymer. So these molecules can form very large gel-like structures. So the next two areas, enzymes, which I'll go into a little bit of depth, primarily covering polyphenol oxidase, but also peroxidase and some of the other oxidase enzymes are important in terms of wheat quality. Alphaamylase is an enzyme that we routinely think about in terms of wheat quality, primarily associated with pre-harvest sprouting, or and as I call them, physiological syndromes. So in terms of quality, an otherwise great variety, if it sits out in the field and gets rained on and the seeds start to germinate and we suffer then what's called pre-harvest sprouting, it can completely ruin wheat quality, even though we started out with a good quality variety. And pre-harvest sprouting is essentially associated or connected with the induction of alphaamylase, which is absolutely necessary for the seedling to get established. We have to break down the starch reserves to fuel the growth of the seedling until it can become photosynthetic and start, you know, making its own food. If that happens prematurely, like I say, it can basically ruin wheat quality. There's another physiological syndrome that you may or may not have heard about. It's referred to as late maturity alpha amylase, and this is actually a developmental defect wherein the regulation of alpha amylase is not under normal control, and if under the right environmental conditions, we can actually have induction of alpha amylase in the maturing grain. Whether alpha amylase comes from LMA or whether it comes from pre-harvest sprouting, in terms of wheat quality, it's almost always a bad thing. The third physiological syndrome that I would point out is something related to, I listed it as heat shock, and really what it has to do is at very elevated temperatures during late grain fill, we can actually get an imbalance of gliadin and glutenin biosynthesis, and it basically throws off the balance of gluten quality, that balance between viscosity and extensibility. So I'll go into PPO a little bit depth now. Here's a kind of a cartoon just showing that whether we have mono or phenolic substrates, tyrosine or L-DOPA, in the presence of atmospheric oxygen and polyphenol oxidase enzyme, those are converted into a intermediary orthoquinone, which polymerize quite quickly into dark colored pigments called melanins, and that's basically the basis for browning and discoloration in foods, whether it's an apple that turns brown or a wheat product that turns brown. Our interest lies primarily in the fact that we're breeding wheat for the Asian market. A lot of our wheat goes to Asia, and a lot of that wheat then would get used in noodles. And certainly the consumer doesn't like a dark or discolored noodle. It's most it's a bigger problem in what we call alkaline noodles, which are actually made at alkaline pH. Just illustrating both the noodle sheets and then the cut noodles here. This is on the left is our noodle sheeter, where we actually take a fairly dry, crumbly dough and sheet it into noodle sheets before cutting. And the lower right is a Minolta color meter, which we would use to actually quantify the color of those noodle sheets. The discoloration, as I mentioned, is primarily a result of polyphenol oxidase, and polyphenol oxidase is nearly all of it is located in a cell layer in the bran, as you can see in the left two panels. And what that does is, then if we look at noodle sheets on the right two panels in greater magnification, we see two phenomena. Certainly we see the speckiness. Each one of those specks is really a tiny piece of bran that's contaminated the flour, as well as a more generalized darkening in the upper panel. There is a tremendous genetic range in polyphenol oxidase in wheat. It's very, very easy to see that if you simply put a dilute solution of phenol on whole wheat kernels. Polyphenol oxidase has a very broad range of substrates, and phenol is one of them. Not that it would normally encounter phenol anywhere in a natural setting. But you can see on the lower right how the phenol is converted into these large polymeric dark, in this case black, pigments. Because of the importance of PPO and the importance of screening wheat breeding lines for low PPO, we developed this test in my lab. It uses L-DOPA as the phenolic substrate. And just to make it easy, we use Eppendorf tubes with 5 wheat kernels. They rotate on this sort of rotisserie mixer for 30 minutes. And then, as you can see in the cuvettes, we would just read the absorbance in a spectrophotometer. More color means more PPO, and we're certainly selecting for lower PPO levels. On the left panel, you can see the wide variety of substrates that PPO will react with. And interestingly enough, they aren't all necessarily black or dark gray colors. Chlorogenic acid in particular gives a, a green reaction, and in general, none of these would be very desirable in terms of a food product. We often group an area of wheat quality traits simply referred to as water relations. And again, it's because generally speaking, when we're making food from wheat, we almost always are mixing it with some level of water. And whether it's a bread dough or a noodle dough or a batter or a cake batter or a tempura batter, the way that wheat flour constituents interact with water is very, very important. So we often list it as somewhat of a separate quality trait. Again, dough-water relations, dough-water absorption is very important. Nearly all bakers would like to have as high a water absorption as possible. They would like to put more water into a bread dough. Water's cheap. In soft wheat products, it's usually often just the opposite. We would like to have the least amount of water in those products because whether you're making a saltine cracker or an Oreo cookie, all of those products to be shelf stable need to be down around 4% or less water content, moisture content. So whatever water you put in to form the product, to form the dough, is going to have to be baked back out. And two things there. One is certainly the thermodynamics of driving out water from a product, and the second is the constituents in a flour that tend to either hold onto or can release water. And the standard these days for breeding programs would be to use one or more of these things called solvent retention capacities. In terms of bred wheat breeding programs, the 10-gram mixograph is probably still the standard. You can see 3 mixograph curves there on the right. Once you become familiar with sort of how these curves are arrived at, you can start to read them, and once you become familiar with their characteristics, you would know that in the topmost curve, that illustrates a very short mixing, very weak gluten flour, and then progressively down in the middle, a medium strength flour, and in the bottom, a very good bread baking strong flour. We can also use something called the farinograph, a very similar, a little bit different mixing operation, gives you a little bit different curve, but still shows the development of the dough and how stable that is to mixing or overmixing. This slide is showing kind of the basic solvent retention capacity test. It uses these 50 ml Falcon tubes. Its flour is mixed with one of the 4 solutions. And then after a very gentle centrifugation, we would drain off the solvent, and we would record the weight, which in a sense is the amount of solvent that has been retained by the flour. And different flours, because of their different compositions, retain different amounts of solvent, hence solvent retention capacity. Excuse me. Lastly, when we get to evaluating breeding lines, really where we think the rubber hits the road, and in a sense it's a summation of a whole bunch of individual traits, is the bake test. Probably in major exporting countries, US, Canada, Australia, Argentina, we're all using some form of a scaled down pan bread for looking at bread quality and hard wheat.
Craig, did we lose you for a minute? Your microphone started breaking up right at the beginning of the slide.
Can you hear me now? Test, can you hear me? According my, huh? Am I on? Okay, thanks. All right, sorry about that. I apparently test. Hopefully you can hear me. It looks like from my end things are working okay. I apologize. Apparently the internet's acting up. Okay. So the last topic that I'm going to cover under sort of the major end-use quality of traits of wheat is really something that's fairly new. Certainly when I got into this business as a graduate student, and even I would say as little as 5 years ago, these are things areas that we didn't really cover. Yeah, okay, so I'll keep talking, and I'm not quite sure what to do about the problems we may or may not be having on the microphone, but if nothing else, you'll be getting the text slides. I do have another mic I can try, I don't know. Let's see, bear with me here for a second.
Yeah, why don't you just keep
Okay, got a new headset. Is that any better? Still breaking up from what I understand. So anyway, it must be the internet connection as opposed to the headset. I will.
And hear you for now.
Okay for now? Yeah.
Yeah, well, we'll post the slides up on the Plant Breeding Training Network. So if there are some times that you fade in or out, you know, people can revisit those spots. Although we're not hearing anything now.
Sarah, you're okay. How's that? Any better?
But yeah, I think so.
Okay. Yeah. For some reason, I have a little blip and I lose my screen and then I also lose my mic. So I'm going to forge ahead. And again, I so I'm going to. Yeah, it seems to be getting worse. I lost my internet connection there for quite a few seconds. I'm going to scroll through the rest of these slides. Again, the mouse picture here is illustrating that we've developed a mouse model for feeding binary mixtures of wheat to mice as a proxy for flavor differences and proxy for flavor detection amongst humans. And some references if you want to, excuse me, do a little more reading in that area. And that's it. And hopefully, I apologize that we kind of spun out there at the end. But whatever you want to do, Sarah, or we can within the limits of the technology here, be happy to try to answer questions or do anything else.
Thanks for your presentation. I thought that was really interesting. You covered a lot of ground. Does anybody have questions here for Dr. Morris? You can type them into the chat box. I had one question I was thinking about. When you were talking about those waxy genes, I was wondering what kind of what the different end uses would be for something that was lacking all three of those genes. What types of uses do you think?
Yeah, yeah. The waxy wheat, what we call full waxy wheat, meaning that it has no functional GBSS and no amylose, is so dramatically different. You almost shouldn't think of it as wheat. It won't make any traditional product, but it also has the potential to make products that cannot be made from any other wheat. So the things to probably start thinking about are products that are puffed or extruded. It makes, I think we're all familiar with Rice Krispies. You can actually make wheat Krispies out of waxy wheat, whereas you couldn't remotely make that out of any normal wheat. So a lot of products like that. Anyway, that's kind of the place to start thinking. The other area that we are, it's quite important in terms of waxy or GBSS, is what we refer to as one gene nulls. So we essentially have two functional copies of GBSS. We've knocked out one of the three genes. So we get a slightly reduced level of amylose, and we get certainly some larger amount of starch swelling. Those sweets are specifically used for Japanese udon noodles, and I don't know how many of you have had udon noodles, but to get that udon noodle texture, one of the things you absolutely have to do is start with these one gene null waxy wheats. Yeah, thanks.
Looks like Mark is typing in a question.
Yeah, it's a huge amount of information, and if you're not familiar with wheat to begin with, it's almost overwhelming. And even if you even if you are familiar with wheat, if you haven't sort of had much experience or dialogue in end use quality, it's just it's a lot of information. And it's true whether you're a graduate student or a postdoc or a wheat breeder. Sometimes I think wheat breeders think cereal chemists are the bane of their existence, but it's the way it goes.
There's a lot of ground to cover for sure. Well, if there aren't any more questions, we won't, hold everyone in here any longer, I guess.
Okay.
But thanks again for your presentation today. That was really great.
Yeah, for sure. And if anybody, let me see if I can get this typed in here. If anybody has any questions or you want to contact me, I'm typing my email address in here. Be happy to interact with anybody. Yep, there it is. So yeah, thanks for listening in.